So we are going to discuss about the internal fertilization, how the internal fertilization is happening. Internal fertilization that is happening in the case of mammals that we are going to discuss in today's class. Okay. So, in suppose here it is the female's uterus and the associated structures. The fertilization occurs in the ampulla region. Fertilization is occurring in the ampulla. Fertilization is occurring in the ampulla region. Before the ampulla, there is a region, you know very well, that region is known as isthmus. That region is known as isthmus. And here this region is known as infundibulum. This portion is known as infundibulum. All these things you know very well. In ampulla only, fertilization happens. Fertilization takes place. Fertilization takes place in the ampulla region of the fallopian tube. Okay. So, when a sperm is coming, when the sperm is coming from the vagina, sperm is coming from the vagina through the uterus, sperm is coming. And when the sperm is reaching near the isthmus, when the sperm is reaching the isthmus, when the sperm is reaching near the isthmus region, when the sperm is reaching near the isthmus region, what will happen, In isthmus region, there is a particular chemical substance that is fibronectin. A chemical substance is present there that is known as fibronectin. Fibronectin is a chemical that is present in the isthmus region. What will happen to the sperm, when the sperm is coming, sperm, when it is reaching in the isthmus region, sperm bind to fibronectin. With the fibronectin, which is high in the isthmus, sperm bind to fibronectin with the help of another protein, with the help of integrin. That is another protein. Integrin is another protein. With the help of integrin, the sperm will bind to the fibronectin, which is present in the isthmus region. Then from the isthmus, what will happen to the sperm, Sperm will undergo. In the isthmus, the sperm undergo, what will happen there, a process that is known as capacitation. So the region where the capacitation is happening, that is isthmus. What is capacitation, After capacitation only, sperm will be able to fertilize. Sperm will be able to fertilize ova. Sperm will be able to fertilize ova only after capacitation, and the capacitation is happening in the isthmus region. From the isthmus, what will happen, from the isthmus? Packet of sperm from the isthmus, packet of sperm, sperm is not going as singly, packet of sperm will be moving, packet of sperm will be released towards, where, towards ampulla, packet of sperm will be released. So with the help of that, the sperm, as the sperm is coming, the total sperm will not go completely. If all the sperms are going completely, if there is no ovum, then fertilization won't take place. So in order to prevent that, what is happening in our body? In isthmus, sperm will be trapped in the isthmus. Then from the isthmus, few numbers of sperm will be passed towards the ampulla. So, all the time, after the release of sperm, for hours, there will be the release of sperm from the isthmus towards the ampulla. It will happen in our body. So, we have to look towards what are the things that is happening during capacitation. So, events in capacitation, we are going to discuss about the events in capacitation. What are happening during capacitation? What are the events that is occurring during capacitation that we can discuss now? The first change in capacitation is change in lipid change. That is the first change in capacitation. Lipid change will occur. This is the first change in capacitation. Normally, suppose if it is a sperm, okay, this is a sperm. Normally, a sperm cell membrane is surrounded by cholesterol. Sperm cell membrane will be surrounded by cholesterol. Cholesterol will be present around the sperm membrane. What is happening, it is happening, during lipid change, the cholesterol is removed. During lipid change, the cholesterol is removed by serum albumin protease. By serum albumin protease. This is a first event. By serum albumin protease, the cholesterol will be removed. Whenever the cholesterol is removed, whenever the cholesterol is removed, what will happen, Suppose if it is a sperm, okay? If it was a sperm, the receptors will be present. The receptors that you know very well, the receptors will be present in different portion. The receptors will be present in different region. Whenever we are removing cholesterol, whenever we are removing cholesterol, whenever there is a removal of cholesterol happen, what will happen, all the receptors will come together. All the receptors will assemble together. All the receptors will come together. So what will happen, all the receptors will become receptors becomes, or receptors come to the tip of the sperm. All the receptors will be coming to the tip of the sperm. So, it ensure what will happen during lipid change, it ensure easy binding to zona pellucida. An easy binding to zona pellucida will occur when there is a lipid change. The second event that is happening during capacitation, that is a removal of protein and carbohydrate. That is a removal of protein. Protein will be removed. Removal of protein and carbohydrate will occur. What is the removal of protein and carbohydrate? The unwanted protein during this step, the unwanted protein, if there is any unwanted protein and carbohydrate, which inhibit unwanted protein and carbohydrate, which inhibit what they are inhibiting, which inhibit the sperm binding to, which inhibit the sperm binding to egg, is removed. That is a second step, removal of protein and carbohydrate. First step was the removal of lipid, that is a lipid change, and all the event is occurring in the isthmus region of the fallopian tube. The third event, what is the third event? That is a change in membrane potential. That is a third event. Change in membrane potential. That is a third event. So all this event is occurring in the sperm. Why all this event is occurring in the sperm? We are making the sperm competent to fertilize the ova, right? So what will happen, during change in membrane potential, what will happen, it will allow calcium channel to open. It will allow calcium channel opening. Whenever the calcium channel opening is happening, calcium enters inside. Whenever the calcium enters, calcium activate. The calcium can activate protein needed for, calcium can activate proteins needed for motility or sperm movement. It can activate the protein, some of the protein which is needed for the sperm movement. That will happen as the third step in capacitation. Okay. So what is happening as the fourth step in capacitation? The fourth step in capacitation is protein phosphorylation. It is a fourth step in capacitation. Protein phosphorylation will occur. What is protein phosphorylation? What is happening in protein phosphorylation? Different proteins present in the sperm, different proteins get phosphorylated. A number of proteins get phosphorylated. Altogether, what will happen, by all these four events? What is phosphorylation? Means addition of phosphate, right? By all these events, all together, the sperm gets activated. All together, the sperm will be getting activated. The sperm gets activated by all these mechanisms. Okay. Whenever there is a lipid removal, we have already discussed about the first change, right? Lipid removal. Whenever there is a lipid removal occurs, whenever there is lipid removal is occurring, it will open, whenever there is a lipid removal, it will open K plus and HCO3 minus channel. K plus and HCO3 minus channel will open whenever there is a lipid removal. So here it is the sperm membrane. Okay, here it is a sperm membrane. Whenever there is a lipid removal, what will happen? One channel will open, that is HCO3 channel. So HCO3 will enters inside the sperm. HCO3 minus will enters inside the sperm. The next channel is K plus channel. So it is a K plus channel. Through the K plus channel, K plus will be removed outside. As we have already told that there is a calcium channel opening. So what will happen? Calcium also enters inside. The calcium will also enters inside. Okay. So what will happen, this calcium and HCO3 minus, calcium and HCO3 minus, calcium and HCO3 minus, when it is reached inside the sperm, when it is reached inside the sperm, that will activate, calcium and HCO3 minus can activate SACY. That will activate a factor, SACY factor will be activated. What is their function, So what will happen, there one ATP will be converted into, cyclic AMP, one ATP will be converted into cyclic AMP. Then with the help of that, that is that will activate pKa. pKa means we have already discussed about pKa. That is protein kinase A. Protein kinase A will be activated there. Whenever the protein kinase A is getting activated, whenever the protein kinase A is getting activated, that will phosphorylate. This protein kinase A only phosphorylate proteins. This will phosphorylate different type of protein present inside the sperm. Whenever it happen, there will be sperm activation. Whenever it happens, there will be sperm activation. There will occur a sperm activation. This is a method by which we are activating the sperm. Why we are doing the capacitation? In order to activate the sperm to fertilize the ova. This is happening. Okay. So listen, whenever the K plus is moving outside, whenever the K plus is moving outside, the sperm membrane, whenever the K plus is moving outside, the sperm membrane will become what, the sperm membrane will become more negatively charged. The sperm membrane will become more negatively charged whenever the K plus is removing outside. We are removing the K plus outside and the HCO3 minus is entering inside, right? So the sperm membrane will become negatively charged. So what is happening next? Sperm is activated now. Sperm is now activated. Now we have to discuss about another thing, that is the hyperactivation of sperm. So what is hyperactivation of sperm that we are going to discuss now? There is hyperactivation of sperm is. So listen, there is when we are looking into this, there is a hyperactivation. We are going to discuss about the hyperactivation. So in hyperactivation, there is increase in, the first one, there is increase in chemosensitive activity. Increase in chemosensitive activities. What is the increase in chemosensitive activity? Listen to me very well. So if you are looking into the ampulla and isthmus, if you are looking into the ampulla and isthmus, in isthmus, in ampulla and isthmus, if you are looking into the ampulla and isthmus, in ampulla, the temperature will be higher 2 degrees Celsius higher than that of isthmus. The temperature will be so this is about the there is chemosensitive activity and another activity is there is increase in thermosensitivity also. Increase in thermosensitivity, we are discussing that together. Increase in thermosensitivity or thermosensitive activities. This thermosensitive activities, in ampulla, if you are looking, the temperature of the ampulla will be 2 degrees Celsius extra than that of isthmus. It will be more than that of isthmus. And if you're looking into the chemosensitivity, if you're looking into the chemosensitivity, what we can say, high progesterone, high progesterone, if you're looking, you can see high amount of progesterone, where, high amount of progesterone in ampulla. So there are two things. There is a higher temperature in ampulla. And there is a high amount of progesterone in ampulla. Due to the presence of this chemical and due to the presence of this temperature, sperm move towards where, sperm move towards ampulla. The sperm will be moving towards the ampulla for fertilization. Sperm move towards the ampulla for fertilization. This will happen. So this is the process that is happening during the fertilization. Okay. So, when the ovum is coming, when the ovum is coming, suppose it is a female reproductive tract, okay? Into the female reproductive tract, who is coming, here, that you know very well. The ova is coming. By covering the ova, corona radiata, everything will be there. Granulosa layer will be there, right? This is known as not ova, it is cumulus oocyte complex. We have already discussed about all these things. It is known as cumulus oocyte complex. Inside the cumulus oocyte complex, we know our ova or secondary oocyte is there. This is cumulus oocyte complex. So the sperm is coming and reaching the cumulus oocyte complex. Okay, sperm is coming and the sperm is reaching the cumulus oocyte complex. This is a cumulus oocyte complex. Sperm is reaching there. What will happen? There will occur the acrosomal reaction. This is an internal fertilization. Okay, there occurs the acrosomal reaction, as like we have discussed in previous class. We have discussed about the fertilization in the case of external fertilization. Acrosomal reaction will occur there. And acrosomal reaction, whenever the acrosomal reaction occurs, these all the granular cells, these granular cells degrade. Granular cells, the granular cell degrade. Granular cell will be degrading from there. Granular cells will degrade from there. Whenever the granular cells degrade, what will happen? Now, the zona pellucida is visible. This is a zona pellucida. Zona pellucida is visible. And the sperm can now reach towards the zona pellucida. Sperm now reach towards the zona pellucida. What is actually zona pellucida? Zona pellucida is a meshwork of glycoprotein. It is a meshwork of glycoprotein. What is glycoprotein? Protein and glucose molecule together. That is glycoprotein. Okay. So these are the glycoprotein, if you are looking in the case of a mouse, you have to buy heart. If you are looking in the case of mice, there is zona pellucida protein 1 is there. Then zona pellucida protein 2 is there. Zona pellucida protein 3 will be also there. Zona pellucida protein 3 will be also there. In the case of a mouse, to the zona pellucida 2 and zona pellucida 3, he is binding to sperm. Here it is a binding position for binding sperm. Zona pellucida 3 and zona pellucida 2 protein, they are binding with the sperm in the case of mouse. Then what is the role of zona pellucida 1? They are stabilizing zona pellucida. The role of zona protein 1 is they are stabilizing zona pellucida. They are only stabilizing the zona pellucida. Zona pellucida 2 and zona pellucida 3 is the binding proteins. Okay. In this manner, if you are looking in the case of mouse, it will be in this manner. Here there will be a zona protein 2 will be there and zona protein 3 will be there. Then zona protein 3, zona protein 2 will be there. Zona protein 2 and zona protein 3 will be there. Zona protein 3 and zona protein 2 will be there. In between them, by connecting this, you can see the zona protein 1. This is a zona protein 1. So into the zona protein 2 and the zona protein 3 only the sperm is binding. And the zona protein 1, what is the role of the zona protein 1? That is stabilizing the zona pellucida. Are you getting me?
Yes, sir.
Okay. If you're looking in the case of humans, if you're looking in the case of human beings, If you are looking in the case of human beings, we are having zona protein 1, we are having zona protein 2, we are having zona protein 3, we are having zona protein 4. These type of zona proteins we are having. So the role of zona protein 1 is as like it is stabilizing, it is stabilize zona pellucida. It will stabilize the zona pellucida. Zona protein 1 will stabilize the zona pellucida. That is the role of this. And zona protein 2, zona protein 3, and zona protein 4, they are the sperm binding protein. They are the sperm binding protein. They are the sperm binding protein. Zona protein 2, zona protein 3, and zona protein 4 are the sperm binding protein. Anyhow, for effective fertilization, if the fertilization want to be effective, then sperm has to bind to where, sperm has to bind to zona protein 2. If the sperm is binding to zona protein 2, what will happen? If it is binding to zona protein 2, what will happen? The fertilization will happen. If the sperm are coming and binding to, if it bind to zona protein 3, zona protein 3, it won't cause fertilization. It won't cause fertilization. Okay, remember, zona protein 2 is the region to which the sperm want to bind. Okay. The zona protein 2, if you are looking into the zona protein 2, that is a region, zona protein 2 is a region between 51 amino acid and 149th amino acid. So that is a region between 51 and 149th amino acid of zona protein 2. There is a binding site. So sperm is binding to zona protein, and more especially in zona protein 2. In zona protein 2, in between 51 amino acid and 149th amino acid, there is a region. In that region only the zona protein 2, in that region only the sperm can bind. Okay. In that region, so sperm has to bind in that region in order to fertilization to be happened. Okay. So here this site, this is a conserved binding site. This is a conserved binding site, This is a conserved binding site in human and rat. If you are looking in the case of human being and also in the case of rat, so that region will be, the binding region will be in between the 51 amino acid and 149th amino acid of the zona protein 2. I hope it is clear. Okay. Now, how the fusion of sperm, how the first sperm is fusing here, fusion of sperm and prevention of polyspermy. Fusion of sperm and prevention of polyspermy. How it is prevented? Fusion of sperm and prevention of polyspermy in the case of human beings or in the case of mammals, that we can discuss. So normally when we are seeing figures and all, a sperm will be coming and sperm will be binding in this manner, right? Sperm will be coming in this manner, right? This 90-degree binding, that is seen in the case of sea urchin. This perpendicular binding, this perpendicular binding, in our previous classes, textbooks and all, we are seeing the sperm is coming and binding in this manner. This perpendicular binding is in sea urchin. In sea urchin only you can see the perpendicular type of binding. Whereas in human beings, in humans, how the sperm is binding, in humans, if you are looking, if it is a ova, the sperm will be binding in this manner. This is a binding in the case of human beings. It is a binding in the case of sea urchin and it is a binding in the case of human beings. In our case, it is side by side. The binding is side by side. Whereas perpendicular in the case of sea urchin. Okay. Suppose it is a sperm, okay, here it is a acrosome, okay, here it is a acrosome. If you are looking, in this junction, if you are looking in this junction, that is, the binding proteins are present, the binding proteins to where binding proteins are present are present here. Binding proteins are present in the junction, in this junction, in this junction, in this junction only, you can see the binding protein. So what is this? This is acrosomal membrane. This membrane is known as acrosomal membrane. This membrane is acrosomal membrane. This is a acrosomal membrane. And what is this membrane? This is a sperm membrane. Sperm membrane. So in between the acrosomal membrane and sperm membrane, you can see a junction. In this junction only, you can see the binding protein. So as the binding proteins are present in this region, a sperm can bind side by side to the ova, not perpendicular, as in the case of sea urchin. Whether it is clear?
Yes, sir.
Okay. Now listen. What are the binding proteins? We are going to discuss about the binding protein. What are the binding proteins present there? What are the binding proteins? The first one, it is about ismo. Ismo is a binding protein. Ismo. What is ismo? Ismo. Ismo is present in the sperm. Ismo will be present in sperm. In the sperm, if you are looking into the sperm, if you are looking into the sperm, here it is a sperm. In this junction, you can see what? You can see the ismo. Ismo will be present here. This is a ismo. So for the ismo, ismo is binding to the ova, right? Ismo is binding to the ova. In the ova, we have already discussed about the ova will form microvilli-like structure that we have discussed in previous class. It will form microvilli-like structure. It will form the structure microvilli-like structure. Over the microvilli-like structure, if you are looking, you can find the receptors for the ismo, the region where the ismo can bind. You can see the region where the ismo can bind. It is known as Juno. This is Juno and this is ismo. So ismo is present in the sperm and Juno is present in the ova. So the reaction is occurring between ismo and Juno for the fertilization to happen. So whenever the ismo and Juno interact, ismo and Juno interact. Whenever the ismo and Juno interact together, whenever the ismo and Juno interact together, what will happen? It will recruit, ismo and Juno interact, it will recruit a CD9 protein. It will recruit a CD9 protein. Whenever the ismo and Juno interact, it will recruit a CD9 protein, and which causes the CD9 protein causes the fusion. The CD9 protein is responsible for causing the fusion in sperm. Now we are discussing about the prevention of polyspermy, how the polyspermy is prevented in this case. Prevention of polyspermy. Polyspermy means only one sperm can only attach the ova. No other sperm, only monosperm is present in our case, right? So there is also slow block. In previous class, we were discussed about slow block and fast block. So what is happening here? Whenever the sperm binds here, whenever the sperm binds to zona protein 2, whenever the sperm bind to zona protein 2, what will happen, whenever the sperm bind to zona protein 2, egg releases calcium. Egg releases calcium from endoplasmic reticulum. Calcium will be released. Whenever the calcium release occurs, cortical reaction will occur. Cortical reaction occurs there. Whenever the cortical reaction occurs, what happens next? Whenever the cortical granule interact, we were discussed about the things what happen after cortical granule reaction. Okay. From cortical granule, from the cortical granule, a chemical will come. The chemical which is coming from the cortical granule is ovastatin. Ovastatin release. Cortical granule will release ovastatin. What will happen, the ovastatin will cleave. It will cleave zona protein 2 from zona pellucida. From zona pellucida, they will cleave the zona protein 2. Zona protein 2 was a region to where the sperm can bind. So what will happen? the zona protein 2 will be removed from there. Whenever the zona protein 2 is removed from there, the further sperm cannot bind there. In previous class in yesterday, we were discussing about the sea urchins case. There were a fast block and slow block were there. Whereas in this case, in our case, only slow block is needed. In our case, only slow block is there. In sea urchin's case, how it was the fast block it was making by changing membrane potential, right? By changing the membrane potential into plus 20 microvolt, they were doing what? They were preventing sperm entry, further sperm entry. But that is not needed here in this case. Why it is so, in the case of sea urchin-like organism, near the ova, a number of sperm will come. In the case of sea urchin-like organism, as it is external fertilization, lot of sperm will come near the ova. Whereas in our case, near the ova, only limited numbers of sperm, because packet of sperms are only released from the isthmus region after capacitation. So only few number of sperm will be coming towards the ova. So there isn't any requirement for fast block. Only slow block is required in our case. Whether it is clear?
Yes, sir.
Okay. Next, activation of egg. Activation of egg. How the egg is getting activated. Activation of egg in our case. How the egg is getting activated. So after the sperm entry, now the sperm is entered. What will happen? Sperm releases. Sperm releases. After the entry, sperm releases, as in the case of sea urchin, sperm releases a soluble compound. Sperm releases a soluble compound. In the case of sea urchin also, we were not knowing the name of that chemical. Anyhow, a soluble compound is released from the sperm. That soluble compound, what that soluble compound will do, that soluble compound will activate, phospholipase C zeta. Phospholipase C will be activated. Phospholipase C will cleave. What they will cleave, phosphatidyl inositol 4,5 bisphosphate. Phosphatidyl inositol 4,5 bisphosphate. I am not writing the full forms and all, because already you know all these chemicals. So from the phosphatidyl inositol 4,5 bisphosphate, when it is getting cleaved, it will be producing what? One IP3 will be released. Yesterday only we were discussed about all these things. IP3 will be released and diacylglycerol, DAG, will be also released from there. This IP3, what will happen, this IP3 will activate calcium. And after that, what will happen, this calcium will activate. This calcium activate egg metabolism. So the metabolism of egg will be starting. Egg metabolism will be starting after the calcium entry. In the case of sperm, in the case of mammalian egg, there is another slow block, and it is caused by Juno protein. Another slow block is there. In our case, one more slow block is there. That slow block is caused by, who is causing, that is caused by Juno protein. We know about Juno. The Juno protein can cause one more slow block. Juno that is present in plasma membrane, very well. When the sperm fuses, when sperm fuses, what will happen, Juno are removed. Whenever the sperm fuses, the Juno will be removed. If the Juno is removed, what will happen? If the Juno is removed from there, then further sperms cannot come and bind there. It is a thing happening in the case of mammalian egg. Okay. Now we have to discuss about the frog oocyte maturation. That is one of the important portion. Frog oocyte maturation that we have to discuss. Frog oocyte maturation. How is the maturation of frog oocyte? That is a topic that we have to study. Frog oocyte maturation that we are discussing here. In frog, yolk will be present in the ova. Yolk will be present. Yellow colored yolk will be there. Yolk provide nourishment. That we know very well. Yolk provides nourishment. In our case, we are having placenta and umbilical cord. So we are getting all the nutrients and all through the umbilical cord. Whereas in the case of this type of organism, yolk will provide the nourishment. So in frog oocyte maturation, the first event is vitellogenesis. The first event is vitellogenesis. Vitellogenesis is the first event. What is vitellogenesis? That is a formation and accumulation. Formation and accumulation. Formation and accumulation of yolk. Formation and accumulation of yolk is known as vitellogenesis. When it is starting, it starts during diplotin stage. It starts during diplotin stage. In diplotin stage itself, the process will start. When the vitellogenesis starts, the oocyte enlarges. The size of oocyte, the size of oocyte enlarges. Oocyte enlarges. The size of oocyte will enlarge. Where the vitellogenesis happens, it happens in the vitellogenesis happens in the liver. It happens in the liver. It happens in the liver and reaches into the yolk formation will happen in the liver and reaches into oocyte. From the liver, that will be transported towards the oocyte. But in primary oocyte, in diplotin stage, in diplotin stage, as we have already discussed about, in diplotin stage, what will happen, in diplotin stage, there is a primary arrest. There is a primary arrest. That means the ova will not develop after the diplotin stage. There is a primary arrest. Because of the why there is a primary arrest, due to the absence of progesterone, due to the absence of progesterone, a primary arrest will occur there in the diplotin stage. That means primary arrest means meiosis will be stopped. Meiosis will be stopped in diplotin stage. So what happened, how the diplotin stage, the stage is getting activated, or the oocyte that will be in diplotin stage. So we have to activate from the diplotin stage. What will happen, what will happen? What is actually happening during the diplotin stage, there is a factor known as MPF. MPF, that is a factor, MPF. That is known as maturation promoting factor. In cell division, we will study about all these things. Maturation promoting factor is there. Maturation promoting factor, that is MPF. This MPF is getting inactivated. MPF is getting inactive. MPF is becoming inactive. MPF is getting inactive due to what? Due to absence of progesterone. Due to absence of progesterone, this MPF is getting activated. This MPF is getting activated. What is present in MPF? If you are looking into MPF, you can see one cyclin will be there. One cyclin you can see. And in MPF, one cyclin dependent kinase will be also there. One cyclin and cyclin dependent kinase. All these are the factors required for cell division. Okay. Cyclin and cyclin dependent kinase will be there in MPF. Okay. So what is happening in adult frog, when it is coming into adult frog, what will happen, when it is coming into adult frog, progesterone will be there. In adult frog, progesterone will be present, right? In adult frog, progesterone will be present. So in adult frog, as the progesterone is present, the progesterone activate. What the progesterone will do, the progesterone activate a kinase. Progesterone will activate a kinase. That kinase is known as CMOS. CMOS will be activated. The CMOS, the CMOS will phosphorylate. The CMOS will phosphorylate whom, the CMOS will phosphorylate P34. The CMOS will phosphorylate P34. Whenever the P34 is activated, our MPF, MPF is activated. Whenever the MPF is activated, what will happen? Whenever the MPF is activated, the arrest is removed. It was in primary arrest, so the arrest will be removed. So it will form, when the arrest is removed, it will form primary oocyte. It will produce primary oocyte. Then to the primary oocyte, what will happen, to the primary oocyte, second meiosis will occur. second meiosis will happen to the primary oocyte. Whenever the second meiosis will happen, again they will go for another arrest. That is known as secondary arrest. Again, they will go for another arrest. Why this arrest? This arrest is due to another factor. This arrest is due to cytostatic factor. Arrest is due to cytostatic factor. That is the next factor. For the secondary arrest, it is due to cytostatic factor or CSF that is responsible for the secondary arrest. What the CSF is doing, CSF that is inhibiting. CSF is inhibiting APC. CSF is inhibiting APC. What is APC? Anaphase. APC means anaphase promoting complex. APC is anaphase promoting complex. APC is anaphase promoting complex. So due to the inhibition of anaphase promoting complex, there will occur secondary arrest. The secondary arrest will occur at metaphase. In metaphase, another arrest will be there. This is a secondary arrest which is happening there. So what will happen next, upon sperm entry, when the sperm is entering, when the sperm is entering into the ova, upon sperm entry, what will happen, upon sperm entry, influx of calcium will be there. Calcium will be entering into the egg. Large amount of calcium will enter into the egg. Upon sperm entry, large amount of calcium will be enters into the egg. Whenever large amount of calcium enters into the egg, the calcium bind to a protein known as calmodulin. Calcium bind to calmodulin. Calcium bind to calmodulin. Calcium will bind to calmodulin. Who is calmodulin? That is a calcium binding protein. Calcium will binding to calmodulin. That is a calcium binding protein. Calcium will bind to the calmodulin. That is a calcium binding protein. Whenever the calcium binds the calmodulin, calmodulin get activated. The calmodulin will be get activated. Calmodulin will be becoming active. The calmodulin activate two protein. It will activate two protein. It can activate two protein. Calmodulin will activate two protein. Calmodulin will activate two protein. Which are those two protein? The first protein it is activating, that is calpain 2 will be activated. Calpain 2 protein will be activated. Calpain 2 protein will activated. The calpain 2, the calpain 2, what is the role of the calpain 2, the calpain 2 that will inactivate, the calpain 2 will inactivate, calpain 2 will inactivate, calpain 2 will inactivate, I hope you remember, CMOS. Calpain 2 will inactivate CMOS. In the previous note, just turn your page, you can see what is CMOS. It will inactivate. Another one is there, another protein is there, that protein is calmodulin dependent. Calmodulin dependent. Another protein is there. That is calmodulin dependent. Calmodulin dependent protein kinase. That is calmodulin dependent protein kinase. Calmodulin dependent protein kinase. Calmodulin dependent protein kinase 2. That is CDPK2 will be also activated. What they will do, they will do what is their role, they phosphorylate. What they will phosphorylate, they will phosphorylate cytostatic factor. I hope you remember CSF, cytostatic factor. CSF will be phosphorylated. Then the CSF is degraded. CSF will be degraded. The CSF will be becoming degraded. Whenever the CSF is degraded, CSF was responsible for the secondary arrest, right? So now the APC, anaphase promoting complex, is free. Whenever the anaphase promoting complex is free, arrest is removed. The arrest will be removed. Secondary arrest will be also removed. Whenever the secondary arrest is removed, what will happen, from metaphase, it was arrested at metaphase stage. From metaphase, they will move towards the metaphase, anaphase, they will move towards the anaphase, and the further division will continue. It is the development in the case of, this is a development in the case of what? In the case of activation of egg in the case of frog. Whether it is clear?
Yes, sir.
Hello?
Yes, sir.
Okay. So, there is a plenty of proteins. Only Elavrasi, Kirta, Shubha, and Shalini is there. A lot of students have informed that they are having examination in their college. That's why they couldn't attend the classes. Okay, no issue. Watch the videos, and I hope you have already studied the topics which we have already conducted in last few classes. Once again, let me record the session first. Stop recording.